Morning, everybody. It's time for lecture number 2 in board review. This is a brand new lecture, so this is actually the first time I've ever given this one, but I did discover a hole in your knowledge when it comes to fetal circulation, especially the exchange of gases at the placenta. So that's kind of the star of the show. And let me get this thing out of the way. I've gotten 0 questions from you guys over the week. Remember, I know some of you are on campus, some of you are off campus, so don't be scared to email me questions if something comes up. All right, are we going to be able, is this going to work? There we go. There's our birds, bald eagle, baby bald eagle. Great, I told you about those, right? So those will be on the test for that's your curve right there. So this is kind of like a cleft palate in a way. This baby bald eagle had a defect in the base of the bill right here. So we could always tell which one that was as it grew older. All right, let's go through this. First time ever presented. Just finished it this morning. This presentation on fetal circulation. So by the end of the 4th week, the 4th week, the fetus has completely developed a circulatory system that has all the parts that the adult does, and then some, as we'll see. So that's pretty amazing right there. It's got a heart that's already pumping its own blood through its closed circulatory system. That's pretty cool. Actually, in week 3, the heart starts pumping a little bit, and you can even at the end of week 3, you can hear an ultrasound detected by, or you can hear a heartbeat detected by ultrasound. But we have a problem, of course. The fetal lungs don't work. They're mature. The kidneys don't work. So therefore, just like any organism, you have a buildup of wastes, and you need nutrients, glucose, and oxygen for cells to stay alive so you can run cellular respiration. We don't have that. So no kidneys and no lungs. So what's the workaround for that? In addition to that, the fetus, or even the embryo, it can't eat, of course. So it has no dietary proteins and fats and carbohydrates. So what's the solution to this problem? Well, mom is the solution. So mom has to provide these functions via her blood. And where does that exchange occur? Where do we get rid of waste and take up nutrients and oxygen, glucose? That's the placenta. And that's an easy thing so far, but we haven't really gotten into how this works, and that's where it gets a little complicated. I could get even more complicated. We're not going down in crazy into the weeds, but you definitely need to know the names of these structures because I have seen them on boards. Okay, yep, so the placenta is the star of the show. And this is the place, there's 2 kind of general parts to it. So there's a mom part to the placenta, and there's a fetal part to the placenta. And then there's a closed space in between here. So the placenta doesn't really function, starts functioning a little bit during week 3, but it's really functioning full power during week 4 when the heart is circulating the fetal blood. So you could say that the placenta takes all the waste out of the fetus or out of the fetal blood, and in exchange for the waste, it puts all the good stuff in, like oxygen, glucose, nutrients. So that's what it does. Let's go a little bit deeper into the placenta. It has 2 parts, and we'll go deeper and deeper into this. Hopefully the board questions won't go any deeper, but they might go deeper than this. So there's a maternal part. That's called the decidua basalis. Decidua basalis. So here's a little cartoon. Kind of rough, but we can see an A to P view of the uterus here. There's the myometrium, perimetrium on the outside. Endometrium would be in here. Cervix down here, fundus on the top. There's our little human growing. There's our little umbilical cord growing. And where implantation occurred Remember the decidual reaction we talked about in embryology? Well, that's where this name comes from, decidua. And this tissue, not the entire endometrium, but the part where the implantation occurs, morphs into this different structure, and that's called the decidua basalis. What did it used to be? It used to be the stratum functionale of the site of implantation, created by the decidual reaction. Great. So that's mom's part of the placenta. All right, and we'll look at subcomponents in a bit. Then the baby, or the fetus, or the embryo, it has a part as well. And this is a new word for you probably. Chorion frondosum. Chorion frondosum. That's the fetal part of the placenta. This is made from the chorionic plate, the chorionic villi. Remember, there's tertiary chorionic villi that we've talked about. I think I'm just getting to that in embryology if you're taking this course at the same time. But it has capillaries. Tertiary chorionic villi is filled with fetal capillaries. So all of that makes up the chorion frondosum. And we can go a step further. The chorion plate is made up of some familiar tissues. It's made up of trophoblast and somatic not intraembryonic mesoderm, but somatic extraembryonic mesoderm. We can go a step further. Trophoblast is really two tissues. It's a cytotrophoblast on the inside, and this cancer-like syncytiotrophoblast. What does that secrete, by the way? What's the big hormone? Great, HCG, human chorionic gonadotropin. So those two make up the trophoblast. So who knows how deep board questions are going to go? So you need to know all this stuff. But at the very basic level, you need to know placenta is made up of maternal part, decidua basalis, fetal part, chorion frondosum. Great. And then in the little cartoon here, you can see that the little kind of windy black cord here, that's the umbilical cord. It connects to the chorion plate of the chorion frondosum of the fetal placenta. See, we can have, like the umbilical cord connects to what? Blank. Placenta. We could go deeper. It detects to the chorion frondosum. Great. We could go even deeper. It connects to the chorion plate or chorionic plate. So there's 3 levels of connection. See how that works. Great. So everything we said already, kind of a review slide. Yep, everything I said. Now, I didn't mention that there's a green closed space here that is filled with mom's blood. That's really important. That's called the intervillous space. Intervillous space is really important. Doesn't have baby's blood. Baby's blood can't get in there. It gets close, but it can't get in there. But that's where mom's blood is. Great. So yeah, intervillous space is between these two parts, decidus basalis, chorion frondosum. Great. And that's the area where gas exchange and waste removal actually occurs. And we'll see, we'll keep going into this here in a second. So now we can see the umbilical cord. So the baby is gone, but we're looking at inside the baby here. So the baby would be coming, or not the baby, but the fetus or the embryo. This looks like the right umbilical vein is gone. So this is after 7 weeks. So let's call this a fetus. But this is the outside world. And this is inside, well, it's not the outside world, but this is inside the baby here. So we're in the abdominal cavity looking out. And we can see the umbilical cord here running off. And great. So the umbilical cord after 6 weeks has an umbilical vein, and it has 2 umbilical arteries. Oxygenated blood is in the umbilical vein. And really, this is the left umbilical vein. As we'll see, I got slides on this. And these are the umbilical arteries. They're moving blood, deoxygenated blood. All right, great. Oh, I set it down here. So prior to week 7, there was actually a right umbilical vein, but that was completely degenerated away by week 6. Those of you who have had my embryology know that premature degeneration of that right umbilical vein is thought to be responsible for the weakness on the right side around the umbilicus, where the intestines can herniate out, which is called a gastroschisis. Gastroschisis, is that enclosed by any membrane, or are those naked intestines sticking out in the wind? Naked intestines sticking in the wind. It does not go through the umbilical ring. It's on the outside of the umbilical ring, but there's no covering. Remember, omphalocele goes through inside the umbilical ring, herniates out, but that's covered by membranes. Great. So let's talk about this PEU, the placental exchange unit. So I don't think you know much about this, and I've seen questions. Actually, I saw one question in my time at the board review on this. So this is where gas and waste exchange really occur. It is the functional unit of the placenta. So the placental exchange unit, or the PEU, that's the functional unit of the placenta, and that's where waste, carbon dioxide, oxygen, and nutrients all swap. Bad stuff goes out of the fetal blood, good stuff comes in. It's made up of 2 parts. So the PEU has 2 parts. It has a intervillous space, which is part of mom. It's not really part of this is not really true. It's not part of the placenta. This is a neutral space between, so I need to change that. It's a neutral space between mom's part of the placenta and the fetal part of the placenta. And here it is. Here it's this hollow part. This goes the whole length of the placenta. That's the intervillous space. So the intervillous space, and then this little tree on the inside. That little tree is the tertiary chorionic villus, which we did talk about in embryology. And so those two together is where this magical exchange of waste and nutrients and oxygen and carbon dioxide occurs. And what's that called collectively? That's called the PEU, or the placental exchange unit. Good with that? Great. How does the blood get in? Because you don't see the blood here, but there's blood flowing around this little tree, mom's blood. Baby's blood is inside this little tree. If we take the skin off the little tree, we can actually see these are capillaries in here. There's no arterioles or venules. These are pure capillaries, and it's just blown up so you can see them. But that's baby's blood. Mom's blood is on the outside. They come very, very close through a membrane here. Syncytiotrophoblasts and cytotrophoblast actually is the membrane, and might as well include the endothelium of the capillaries. So I think I have a slide on that coming. But there it is. There's the umbilical cord attaching to the umbilical plate here. And yeah, we can see that. Here's where the small arteries and veins, and they become arterioles and venules. They're the ones that go into this little tertiary chorionic villus. But remember, baby's blood is in here. Mom's blood is outside. Where's mom's blood? It's in the intervillous space. Where's baby's blood? It's inside this tertiary chorionic villi, not a secondary. Remember, that's got mesoderm, or that's got mesenchyme in it. Certainly not a primary. That hasn't formed anything yet. Spiral arteries of mom, where do they dump blood? They don't connect to these little trees, right? These tertiary villi don't connect directly with mom's spiral arteries. They dump blood into this little space right here, again called the intervillous space. Good with that? All right. I see you shaking your heads. You can probably figure that out. So there's another concept of a placental membrane, or a placental barrier, that this is where the exchange of waste and nutrients occurs. Who is, or where is this placental membrane? It's the walls of this little tree, including the walls of the capillaries. That's the placental membrane. Okay, so who are the walls again? Going from outside to in, and I don't have, like I said, I just finished making these slides this morning, so eventually I'll get a blow up of this and I'll show you the layers. But going from outside to in, cytotrophoblast, then we have, the very outside layer is syncytiotrophoblast. The next layer down is cytotrophoblast. There's a basement membrane too, we won't worry about that. And then you're going to hit the endothelium of the capillaries. So those are the 3 layers. Ooh, 3. I like to make questions on that, don't I? The placental membrane has 3 layers. Syncytiotrophoblast, cytotrophoblast, and the capillaries endothelium. That's called fetal capillary endothelium. Yep, remember, trophoblast splits. It kind of evolves into 2 layers. Cytotrophoblast and syncytiotrophoblast. Great. Another fact over here, this intervillous space, where is it between? That's this whole space right here. It's said to be between mom's part of the placenta, which is the decidua basalis, and the chorion chondrosum, which is the fetal part of the placenta. It's a space in between. It doesn't belong to either one of those placentas. And yes, the intervillous space is filled with not the fetus blood or the embryo's blood, or however the question will come at you. It's filled with mom's blood. Good with that? So it's kind of a similar story, right? These almost look like alveoli in the lungs, pulmonary alveoli. The only difference is in pulmonary alveoli, we're exchanging our human blood, dumps oxygen or dumps carbon dioxide, some hydrogen, things like that, into the air. But here we have blood dumping the same stuff, hydrogen, carbon dioxide, urea, bilirubin, all that stuff, but it's dumping it from the fetus blood into not the oxygen, but into mom's blood. So it is similar to a pulmonary alveoli. Again, mom's maternal blood and the fetal blood never normally touch one another. Now, during delivery of the placenta, which is huge, I should have put a picture of one of those in there. But it's a big structure, and sometimes you can get a tearing of the placenta, which is not good, because then if you rip the structure open, baby's blood and mom's blood can mix together. The baby's usually taken out by then, but mom can be in trouble if she's got the wrong RH compatibility with that blood. You can kill her. So that can be dangerous. Another lecture on RH. Maybe we'll get to that one. Okay, let's see. How does waste and nutrient exchange work? Well, we kind of said that already. Mom's blood, not really shown, is pumped into this intervillous space. Great. Where does it come from? Spiral arteries. Definitely know where the spiral arteries come from. The bad blood, or kind of the blood that's filled up with waste products, is drained through maternal veins. You can see the little ostium there. Great. Fetal blood is pumped into the tertiary chorionic villi. So where does the umbilical cord ends at this plate, chorionic plate, but the vessels come out of it and go all the way into these little tree-like structures, tertiary chorionic villi. Yeah. Two bloods come very close, but they don't contact one another. We've said that. The membrane of the tertiary chorionic villi is where gas exchange and waste removal occurs. We said that was the what? We said that was the placental membrane, or the placental barrier. All right, I think we got that slide down. Yep, again, mom's and baby's blood don't mix. Pretty much said everything about that. All right, great. Are we good with that? I know that's probably new for you. Maybe not, but let's bring that back to the surface, because I did see that on a real board test. Fetal circulation. Let's get into that. So we already said the fetal circulation is already developed by week 4, ready to go, but it's unique. it's got a couple extra parts to it we need to talk about. And a complicated little area is when it gets close to the liver. Fetal circulation actually diverts blood away from two structures. From an immature liver, puts a little blood into it, but most of that's reclaimed, and into the lungs, which aren't functioning. So therefore the anatomy is a little bit different here. Let's see. So we looked at this already. The umbilical cord carries oxygenated and deoxygenated blood. I think we know that. We said that the umbilical vein is the one that's heading toward mom's heart. So it does have oxygenated blood in it. And this is really the left umbilical vein. So this picture is drawn after the 7th or after the 7th week, because we don't see the right umbilical vein, just one left. And we said the right umbilical vein degenerates about week 6. And we said that gastroschisis can occur if it degenerates away too early. Great. Anybody see anything wrong with this picture? Actually, I put this into chat. I've paid Chat GPT now, and Perplexity. Perplexity missed this one. I wanted to see if these AI search engines could catch the error on this slide. So the error is right here. So there's the round ligament, the obliterated umbilical vein. So this left umbilical vein, after the fetus is born, becomes a baby, this obliterates into nothing in the falciform ligament, in the bottom, the free edge of it, and it becomes the round ligament of the liver. But the problem here is it's in two places at once, so the author completely screwed this up, and the time is way off. So still a pretty cool picture though, but that's definitely shouldn't be there. Okay, rounding the liver. This was a tough story. It took me a couple hours to get this down. I had to check. Perplexity screwed this up, the references, but ChatGPT actually got this right. I always check references with these AI search engines. Yeah, definitely now ChatGPT, that $20 a month, is an amazing search engine. It'll search the board books. I don't know how it gets around it. It'll search the board books and give you the chapter and the page, and they're usually pretty close. They're usually a version older, like an edition older or two, but really amazing study tool if you're not using that. Got to be careful not to get bogged down in it, because you can fall in rabbit holes really easy. But for professors, it's really good for helping with tough areas. Anyway, so the liver story. So here comes our left umbilical vein. It's traveling in some mesentery, some ventral mesentery. Perplexity said it was traveling in the falciform ligament, which is crazy because the falciform ligament isn't formed at this time. ChatGPT got this right. So it's traveling in some ventral mesentery, kind of wrapped up. And when it comes to the liver, it's arguable what happens. The anatomy can be quite variable here. But one key thing happens. It does enter the liver, and it ends and gives rise to the ductus venosus. Remember ligamentum venosus in the liver in cadaver lab? This is where ligamentum venosus came from. Right now it's ductus venosus. It takes 50%, approximately 50% of this oxygenated blood, and it straight runs through the substance of the liver and dumps it into the vena cava here on the superior part of the liver. Nothing is lost here. However, so you can see in this story, and this can vary. Sometimes this can dead into the left portal vein. Sometimes the portal vein can dead end into it. But the bottom line is some of this blood, about 50% of this blood, ends up going through the left hepatic sinusoid. And the hepatocytes live, they're not drawn here, but the important hepatocytes, they make albumin, all the clotting factors, super important, right? They live in here, and they use this sinusoid, which is a sinusoidal capillary, very, very leaky. So about 50% of the blood goes through ductus venosus, 50% goes through this left hepatic sinusoid, and it does oxygenate some of the tissue here. The right side, for whatever reason, usually doesn't get this good oxygenated blood, which is kind of strange, but it doesn't. And then it dumps back in. I think there's a hepatic vein it goes into. This is hepatic vein, and it dumps into the inferior vena cava. But the key is when this blood comes back, it's lost about half of its oxygen. So the deoxygenated blood mixes with mom's 100% oxygenated blood, and now we got a blood that's not 100% oxygenated. Right? That's the story with that. Everything I said about the ductus venosus, that's the key thing. What does that become in the adult? Ligamentum venosus. Just like there's a ligamentum arteriosum, there's a ligamentum venosus as well. Okay, everything I said. Great, left hepatic portal vein. The board books say that this, I couldn't find a drawing of it. I didn't have time to Photoshop it, but the board books say it kind of dead ends into this left portal vein inside the liver, and then the left portal vein gives rise to the left hepatic sinusoid, and that's how it goes. So obviously I'm not going to hit the details. The big detail that's going to be hit, though, is that this ductus venosus has 100% oxygenated blood, even though it passes through the inside of the liver. It's not on the outside of the liver. It just doesn't connect to anything. All right, great. We've done this already, so you're experts in the heart. So the only thing I wanted to bring back up, and you're responsible for all this, we talked about this in Lecture 1, but there's a hole here, and that's called Foramen Ovale. After you're born, this little flap, which is called the valve, is pressure closed against the limbus. I don't think I talked about that yet, but we will talk about that. But yeah, so that's open. We also have that little guiding fin. There's the eustachian valve. So mom's blood, oxygenated blood, will come up through the inferior vena cava, and it'll hit this fin, and the fin will guide it right through this hole, and it'll get into the left side of the heart. That's the part there. Remember the AV node. I think a practice question asked about where the AV node's located. It's at the tip of the triangle of Koch. Some call that triangle of Cook. Great. Remember the terminal crescent is what gives rise to pectinate muscle. Great. Remember both auricles are filled with pectinate muscle, but the left atrium does not have pectinate muscle except in the auricle, and that's where blood clots can form in people who get atrial fibrillation. Great. Oh, we definitely know that too. Fossa ovalis in the adult heart is made up of a limbus and a valve, and that valve can flap around. Normally it gets welded shut within the first years, we'll see. All right, so everything I really said before, mom's oxygenated blood comes in, hits the eustachian valve, which guides it through the foramen ovale. It's pink. Why is it pink and not pure red? Well, it's mostly oxygenated here, although we know it's not 100% oxygenated because of that trip through the hepatic sinusoids. But we'll just say it's almost purely oxygenated here, but it's hitting the superior vena cava's deoxygenated blood is coming down and mixing a little bit with it. And so we don't have red over here anymore. We have kind of a pink showing that it's lost a little bit of oxygenation. But nevertheless, it goes through here, and now it shows up over here in this kind of cool little heart that's not really right. The left atrium is usually not seen. Left atrium would be right here, but into the plane of the page. But anyway, another point we need to talk about is that not all the blood goes through this hole. So some of this pink blood misses the hole, and there's a current coming down, and there's an atrial systole, and some of the little blood actually ends up, the deoxygenated blood. Not deoxygenated, I don't want to say that. It's still got quite a bit of oxygen. It's still usable. It gets in the right ventricle. Unlike the adult, this is not usable. It's deoxygenated. But in the fetus, in embryo, it's it's usable. So if we can reclaim it, that's great. Let's look at the left side first. So left side is normal. The ventricle fills up, filling phase of the cardiac cycle, and then we get atrial systole, fills the ventricle 100%. We'll talk about that later. The ventricle contracts, and all that beautiful blood goes up around the horn. Great. Just as normal, goes down the thoracic aorta, descending aorta. Wonderful. But what about this deoxygenated, or this still oxygenated blood? That we need to reuse that. So how does that work? And that's where the ductus arteriosus comes into play. So we have another little tube. It's just like the ductus venosus. The author didn't show much of a tube here, but there is a little tube that can be quite long, and it connects at the pulmonary trunk, where the pulmonary trunk splits into the pulmonary arteries. There's usually a little, or there's always a little tube here that connects to the part of the thoracic aorta right after the aortic arch. This one's off a little bit. It should really be right about here. And so what's the story? Why do we have a hole right here? So we have blood flying around here, and now we have a hole. Well, some of the blood that's during systole, the blood is going to be blasted up here. Some of the blood goes right through this hole and is reclaimed in the, what's called the aorta blood. Some of the blood blasts down toward the lungs, and the lungs are closed for business. Not much blood can get through, a little tiny bit. So very high pressure, it's like a beaver dam in the lungs, so we get high pressure here. So when systole occurs, this blood flies up the right ventricle, up the pulmonary trunk, and it hits this high pressure zone, and it goes, whoa, we're not going down this way, and it's diverted into this hole. And therefore, the descending part of the aorta right here, it receives blood not only from the ascending and aortic arch, it also receives some blood from the ductus arteriosus. So that's the story with that. And we'll talk about what happens when that doesn't close. Great. So everything I said already, you can read through that. Great. Yep. So the blood is the pulmonary arteries, tries to take it, but it's closed. It makes very high pressure there. And so the blood is shunted through the ductus arteriosus. Great. This area right after the aortic arch actually has a name. That's called the isthmus of the thoracic aorta. Where's the isthmus of the thoracic aorta? It's just downstream of the aortic arch. And that's the place where the ductus arteriosus and the ligamentum arteriosus are found, in the isthmus of the thoracic aorta. You've heard of coarctation of the aorta? That usually occurs in the isthmus of the thoracic aorta as well. Okay, great. So we got blood circulating, going around the horn. It's going off into all the tributaries here: brachiocephalic trunk, left subclavian, or left common carotid, and then we have left subclavian. You should know those, of course. Brachiocephalic veins. Anyway, it's going around. So the head is supplied with full power oxygen, the best we can do. Great. The arms are supplied. But it turns out that as the blood turns and start going down, it gets robbed really quick of oxygenation. We got the celiac trunk, takes a lot of the oxygenation out of it. Superior splanchnic artery steals some. Inferior splanchnic steals some. We got the renal arteries is all stealing this blood, and it turns out by the time it gets down here into the common iliac, specifically into the internal and external iliac, the blood is like 50% of the oxygen is gone. It's really not super useful. So the committee decided to get that back and get that blood recharged at that point. And so umbilical arteries, which we don't have in the adult. We have remnants. What are the remnants of the umbilical arteries? Quick, quick, quick. Right and left medial umbilical ligaments. Remember, there's a median umbilical ligament or fold. No, they're just ligaments. And there's a right and left medial umbilical ligaments. Those are the remnants of these guys. But anyway, so our blood comes down here, and the committee goes, let's recycle this blood. And so we got these vessels going, goes through the umbilicus, goes through the umbilical cord with the skin removed here. And yeah, it goes back down to the placenta, goes into the tertiary chorionic villi, and the whole story repeats again. So the obvious weird part about this is, how's the circulation, or what kind of perfusion do the lower extremities have, and the pelvis really have, compared to the upper extremities and the heart and the thorax? Well, they kind of get cheated out of blood. So extremities, feet, legs, thighs, they don't get as much blood. They only get 50% oxygenated blood. So, but I guess that's fine. It works out okay. The good oxygenated blood is saved for all the organs and the head and the arms. So that's kind of weird part of the story. Okay, yep. And then when we come back down, here comes our the right and left umbilical arteries. You can see them each dumping into this little Christmas tree, which is tertiary chorionic villi. And then the story repeats, and we get dumping off of that waste products. When those waste products get dumped into mom's blood, they stream right into the maternal veins. Because I was wondering, I asked GPT that question to see if it could research. I was wondering if some of the creatine and some of the waste products, the CO2, could that possibly get sucked back up into the arterial circulation? They said no. The board books say it just streams, it flows directly into the vein. And they're a little shaky on why that is, but it doesn't get taken back up again. All right, great. Yep, so there's the remnants. So if we look at the anterior abdominal wall from the inside out, belly button would be right here. So this is a P to A view from the inside the abdominal cavity. We can see the remnants right here. Those are the right and left medial umbilical ligaments. What's that thing in the middle? That's the urachus. And there's some non-remnant structures. Those are the lateral umbilical ligaments, which house the inferior, I believe it's the inferior epigastric artery. It's a little peritoneum envelope that they can travel in, but that's not a remnant of anything like the other two are. What's the urachus a remnant of? The lantois is probably the deepest answer. Lantois. Oh, and then the urachus, and then we get the median umbilical ligament. Did I say that? Urachus is derived from urachus. Urachus is derived from the lantois. What happens in the heart? We already know this, so we don't have to talk about that again. But the key is that fossa ovalis. So let's go a little bit further. I can't see my clock. I don't want to make this like too much of a lecture. Fossa ovalis, we know about it. Turns into the foramen. Or fossa ovalis was the foramen ovale. After the child is born, the valve pressure closes, and it's no longer a hole. There's a depression there, so it's called a fossa ovalis. It's very thin. You can shine a little pen light through it. You know, the light that you test the light reflexes for. You can shine that right through there, and you can see the light on the other or in the other atrium. So it's between the atrium. It's part of the interatrial septum. Used to be foramen ovale. Pressure closed at birth. Permanently closed in about 80% of us at 1 year. Pressure closed. What the heck does that mean? Well, let's look. You can read about it right here. Let's just look at the picture. So once you're born, the lungs start working. right? You lose the umbilical cord, everything starts working, and the lungs start dumping lots of blood into the left atrium, and blood is pumped through the left ventricle and it's shot out. The left side of the heart is very muscular and powerful, so when it contracts, the pressures are way higher compared to the wimpy right side of the heart. When it contracts, the pressures aren't that high. So when the atria contracts, that high pressure, it keeps a finger on this. It pushes against this valve of the fossa ovalis, and it holds it there, and it pressure closes it. Even the blood coming in to the left atrium, even if we didn't have atrial systole, that blood is under much higher pressure compared to the blood that's coming from the inferior and superior vena cava. That's like 3 millimeters of mercury, and this is much, much higher, like 25 millimeters of mercury. So therefore, high pressure, low pressure, some of the blood tries to go this way, but it's stopped by this valve, and that's what that means. So high, beautiful oxygenated blood, 100% oxygenated blood, it's kept out of the venous blood, which is, maybe got 5% of oxygen in it. So great, that's how things normally work. And then if we go back to this, because this is held constantly, this kind of fibroses shut. It welds shut. So in most humans, I think it's about 80% of people by the end of 1 year of life, this is welded shut. So if you ever do get high pressures in the right side of the heart from disease like COPD or some stenosis of the pulmonary valve or tricuspid valve, you can get very high pressures here. It'll push, but this won't open because it's been welded in those 80% of people. So actually it's worse. 75% of humans get that valve welded shut. So that means that that means that 25% of humans don't have that welded shut? It's like, what the heck? Yep. 25% of humans, it doesn't weld shut. 75% of humans, it does weld shut. But there's people listening to this right now who have a freely movable valve of the fossa ovalis. You'll probably never know because you'll probably never get COPD or any trouble. But if you do when you get older, that can be problematic. So what if the weld doesn't happen? This is the probe patent foramen ovale. So I was going to save this for next week, but we're doing pretty good with time. Or are we? I forgot what time I started. Let's go just a little further and look at this. Maybe we'll review this when we get going next time. So about 25% humans, the valve and the limbus never fibrose together. This is called a patent foramen ovale or a probe patent foramen ovale. Doesn't mean it's symptomatic, but it means it's there. It's a congenital defect. Some people argue about that because it really shows up after birth. So some people say it's acquired, but most say it's congenital. And there's a regular closed limbus and valve, and there's one that's somebody's sticking a probe through, and the early cardiologists called it, might as well call it a probe patent foramen ovale, and that's where that came from. So in people with normal pressures, who cares? It's pressure sealed. Now there's some talk, you probably talked about this in CVP class, but sometimes you can get occasional very high pressures, like if you're doing squats at the gym, or you're on the toilet and you're bearing down real hard, you're constipated, or you're giving birth. You can get pretty high pressures in this side of the heart, enough to maybe pop this valve open. And the problem is if you're pushing and you happen to dislodge a blood clot that's in the lower extremity vascular, or in the veins of lower extremities, I should say, dislodge thrombus formation and cause an embolism. If you're pushing at the same time, it's possible to push the embolism over into the place where it should never go, into the left side of the heart. Because from the left side of the heart, just think of the places the blood can flow. Up into the brain, you could get a stroke. Back into the heart, you can get a heart attack. up the celiac trunk, you could lose your liver and your stomach. So that's that could be problematic, but for the most part, people don't have trouble with that. So you won't shunt. Okay, everything we said. But if you do ever develop pulmonary hypertension, probably from COPD, or it could be from a stenosis or a sticky tricuspid valve or a sticky pulmonary valve that doesn't open well. That can raise the pressures on the right side of the heart. But if they raise, then you're going to get a shunting of blood in the wrong direction. You'll get a pathological right to left shunt. So people with a PFO, if they ever develop pulmonary hypertension for whatever possible reason, it usually comes from left heart failure. But it doesn't matter if you develop pulmonary hypertension and the right pressures in your heart go above that of the left pressure, you can get that nasty deoxygenated blood leaking through the old Foramen Ovale, which is open now, probe-patent Foramen Ovale, into the beautiful oxygenated blood in the left atrium, and the body does not tolerate losing the oxygenation of blood. The body does not tolerate the mixing of deoxygenated blood into oxygenated blood. And you'll start turning blue, you won't feel good, you can't climb stairs, you'll become very sick. If you have a hole in your heart and you lose some of the oxygenated blood into the deoxygenated blood of the right heart, no big deal for a while, but it's not good for your lungs, right? You probably learned in CVP how it wrecks your lungs and causes pulmonary hypertension. But yeah, so here's someone with COPD. They have chronic bronchitis, predominant COPD. They have a little bit of emphysema as well, right? You can never have pure emphysema or pure chronic bronchitis. They're always mixed together. COPD. So the pressures are really high right here, and we can see that that the high pressure is forcing blood into the left atrium, and it's wrecking it. It's taking the oxygenation away. So now you got a pump of blood that's maybe 50% oxygenated, because this nasty blood has shunted into it, and you're going to be really sick from that. See how that works? Yeah, so beaver dams anywhere to the outflow of the right heart. So we could have a beaver dam here. We could have an embolus, like blocking this, a saddle embolus. We learned about those in CVP, I hope. That could cause it. So the blood's trying to get this way, but it's traffic jam, right? It's a beaver dam. So the blood backs up and the pressure raises. I could have put one here. I could have put one here, tricuspid and pulmonary valve, if they're not working. Anything that pressurizes this right atrium will push blood through a opened valve, and you will wreck your oxygenated blood in your left heart. Good with that? All right, what are the causes of pulmonary hypertension? Well, we mentioned some of them. COPD for sure. Pulmonary embolism, stuck like right at the junction, or right where the pulmonary trunk splits into the pulmonary arteries. Heart failure, probably the number one cause of right heart failure is left heart failure. It's a beaver dam in the left heart. The blood backs up all the way through the lungs and all the way into the right heart, increasing pressures. Valvular stenosis on either side, the aorta, on the left heart or on the right heart. Any valvular disease can do it. Tumor crushing the proximal pulmonary veins, any type of beaver dam. See how that works? Congenital heart disease, so a hole in the heart. So if you have a hole in, let's say, your intraatrial septum, and on every beat of the heart, extra blood is squirting into the right side of the heart, that extra blood will wreck the microcirculation of the lungs. And pretty soon, the lungs will be scarred up, and it's harder to push blood through the lungs when they're scarred up. So they become like a beaver dam, and the pressure backs up. So that can work as well. There's some idiopathic pulmonary hypertension where they can't figure out why the patient's developed pulmonary hypertension. One theory is that the endothelial cells in the lungs oversecrete something called endothelin, which causes vasoconstriction of the microcirculation of the lungs, acts like a beaver dam. All right, I think that's probably enough for your brain. So we kind of said this already. So the dangers of a right to left shunt. Did I say left to right before? The danger is going from right atrium to left atrium. I have to run back and look, but the danger is right to left shunt. Left to right shunt's not dangerous. It could cause Eisenmenger's syndrome, but it's not, and eventually cause increased pulmonary pressures or increased pressures in the right side of the heart. But it's the right to left shunt that is so dangerous. But yeah, we talked about a paradoxical embolism. If you have pulmonary hypertension, then the blood is always flowing that way, and you get thrombus broken loose. It causes an embolism, goes up through the heart. Normally it would just go and get stuck in the lungs, but if there's a hole, it could get pushed into the left side of the heart, and you could die from a stroke or heart attack, or as we said before. Let's see. Yeah, arterial septic emboli, so the non-laminar flow through like a PFO is a sticking place for bugs. When you're not supposed to have blood flowing through a little hole, so if you get bacteria in your blood, it can stick there and grow there, and you could get a bug ball embolism that breaks loose and causes trouble. Yeah, I think we said this. So PFOs in patients with right to left shunts are at risk for venous thromboembolism into the left side of the heart. That's called a paradoxical embolism. I think I have a picture of that somewhere. Yeah, because it's dangerous, right? Just think with the left side of the heart, where does that blood go? It goes up into the brain. It goes everywhere. Here's the cartoon. So this person has COPD, and they have high pressures in the right side of the heart. And so some of the blood is always flowing through this patent foramen ovale, or patent fossa ovalis, whatever you want to call it. And bad luck, an embolism breaks loose and it's traveling. It should go into the lungs, but the timing is just perfect. Atrial systole occurs to make things worse, and this big old clot goes through the foramen ovale, or the open fossa ovalis, and now it's on the loose in the left side, and you can use your imagination where that thing goes, right? If you come out of the left heart, look at all the places you can go. You can go this way, get up in the common carotid artery. You can go up the left common carotid artery, go into the internal carotid arteries, go up into the brain, into the circle of Willis. Got yourself a stroke. So, yeah, not a good thing. Yeah, so they could get stuck in anywhere. We just said this: coronary arteries, cerebral arteries, celiac trunk, renal arteries, splanchnic arteries, and they lead to all this sequelae. All right, good place to stop. I'll put some, if you haven't seen, there are some book practice questions up from week 1. They're in the little tile. I guess it's called a widget, but it's a tile, I think is a better name for it. So I'll get some up for this week's lecture as well. Email me if you have questions. See you later.